press the bell icon on the YouTube app and never miss another update.
Hello everyone. So this is my third video on the 8th chapter, Cell, the Unit of Life. In this video we are going to study the endomembrane system. Endomembrane system. Endo is inside, membrane system. Hope you all understand what is membranes. So the cell contains in it so many membranous structures. The other day we had classified membranous organelles and non-membranous organelles, single membranous and double membranous organelles. So the fluid that is present between the plasma membrane and the nuclear membrane is nothing but the cytoplasm, in which are the embedded structures like the organelles. Among them, some of them are a part of the endomembrane system, and the part of the endomembrane system are the endoplasmic reticulum, the Golgi body, lysosomes and vacuoles. Their functions are closely related, and therefore they are also called as they are collectively called as endomembrane system. But whereas the mitochondria, peroxisomes, plastids, their functions are not related with the functions of these organelles, hence they do not come under the endomembrane system. So the endomembrane system in the cell is made up of endoplasmic reticulum, the Golgi body, the lysosomes and the vacuoles. So let us start now our study with endoplasmic reticulum. The endoplasmic reticulum is simply referred as ER, or it is also called as ergastoplasm. It is also called as ergastoplasm. The endoplasmic reticulum was discovered by scientists called as Porter, and these endoplasmic reticulum, which means endo is inside, plasmic is which is the one present in the cytoplasm, and the reticulum is nothing but the network. So these are ultra microscopic network of interconnected membranes membrane made of cisternae, vesicles and tubules. So these are network of interconnected membranes made up of cisternae, vesicles and tubules, which are present in the cytoplasm, and they are ultra microscopic. So they form the endoplasmic reticulum. They are somewhat connection between the plasma membrane. They are somewhat the connection between the plasma membrane and the nuclear membrane. So they are found scattered in the cytoplasm, hence they are called as endoplasmic reticulum. So these endoplasmic reticulum, they divide the intracellular space. They divide the intracellular space into luminal space and extraluminal space. So they divide the intercellular space, the space within the cell is divided into luminal space and extraluminal space by the endoplasmic reticulum. So these endoplasmic reticulum, they are found as three components. There are three components of endoplasmic reticulum. One is the cisternae. The second is the tubules, and the other is the vesicles. I shall write the diagram also. So cisternae, or they are flat sac-like structures. They are flat sac-like structures present one above the other. They are flat sac-like membranous structures which are placed one above the other. Flat sac-like membranous structure which are present one above the other. And these tubules, they are branched, elongated, tube-like structures. Tube-like. And the vesicles, they are rounded structures. So they are spherical structures. These tubules are also branched. The cisternae, the diameter of the cisternae are about nearly 40 to 50 nanometer, and the diameter of these tubules is in between 50 to 100 nanometer, and vesicles is about 25 to 500 nanometer in diameter. So cisternae, they are flat sac-like structures which are placed one above the other, and they are membranous structures. The tubules are elongated tube-like structures which are also branched, and they have a diameter of nearly 50 to 100 nanometer. Vesicles, they are spherical structures, so they have a diameter of 25 to 500 nanometers. Now looking into the diagram of this endoplasmic reticulum, so these are the cisternae. So coming to the tubules, they are elongated structures tube-like structures which are branched, and the vesicles are rounded structures, almost spherical structures, where the diameter is in between 25 to 500 nanometer based on different locations they are present. So these are the three different components of the endoplasmic reticulum. So these endoplasmic reticulum are sometimes studded by the ribosomes. They are studded by the ribosomes. That is, the ribosomes are placed on these membranous organelles, that is the endoplasmic reticulum, thus making the surface rough. So if they are naked without ribosomes, you call it as smooth ribosomes, and these are nothing but the rough endoplasmic reticulum. The rough endoplasmic reticulum. So if we simply write structure without, right here. So without these ribosomes, it might be smooth endoplasmic reticulum, but if it is studded with ribosomes, then it is called as rough endoplasmic reticulum. So there are ultra microscopic network of membranes structures which are made up of cisternae, vesicles and tubules and are present in the eukaryotic cells. They are present only in the eukaryotic cells and absent in case of prokaryotes. All eukaryotic cells except matured RBC, except the matured RBC, they are present in almost all eukaryotic cells. You know, the matured RBC doesn't contain the nucleus. Along with that, it will not have the cytoskeletal structures also. So they are called as the cytoskeletal structures. That means they form the skeleton of the cell and they give the mechanical support to the cell. Hence, they are also called as cytoskeletal structures. The endoplasmic reticulum is called as the cytoskeletal structure. Then they are the connection between the plasma membrane and the nuclear membrane. So the intercellular space, that is the space inside the cell, has been divided into luminal space and extraluminal space because of the distribution of this network of tubules inside the cytoplasm. And these endoplasmic reticulum, they are made up of three different types of components: the cisternae, tubules and the vesicles. So the diagram is like this. So they are flat sac-like structures which are placed one above the other, and these are branched elongated tube-like structures, and these are rounded vesicles. So the diameter is also mentioned over here. The cisternae have got a diameter of 40 to 50 nanometer. The tubules have a diameter of nearly 50 to 100 nanometer, and vesicles may range from 25 to 500 nanometer in their diameter. So now, rough and smooth endoplasmic reticulum. Rough and smooth endoplasmic reticulum. The rough endoplasmic reticulum, rough endoplasmic reticulum, and it is referred as RER, and the other is the smooth endoplasmic reticulum. Smooth endoplasmic reticulum. So it is referred as SER. It is referred as SER, RER and SER. Rough endoplasmic reticulum, the surface of the endoplasmic reticulum is rough due to the presence of ribosomes. And some of the examples of these RER containing cells are the goblet cells, the plasma cells and the fibroblasts. So these are different types of cells. I don't know whether we have come across these cells or not. Goblet cells or the secretory cells. The plasma cells are the cells which are concerned with the immune system, and they form the antibodies in our body. So the plasma cells, they form the antibodies, and the fibroblast, they secrete fibers. All of them almost they are secretory in function. The goblet cells, the plasma cells and the fibroblasts. So smooth endoplasmic reticulum, they are the one which the smooth endoplasmic reticulum, wherein the surface is smooth due to the absence of the ribosomes, and the examples are adipose cells, liver cells, leukocytes and also adrenocortical cells. So adipose cells are nothing but the fat containing cells, and liver cells, we know them, they have the hepatic cells, cells belonging to the liver, and the leukocytes are nothing but the WBCs, and the adrenocortical cells or the cells present in the cortex region of the adrenal gland. Cells belonging to the cortex region of the adrenal gland, they are all examples where the smooth endoplasmic reticulum is present. Now coming to the functions of endoplasmic reticulum. Coming to the functions of endoplasmic reticulum. So, very firstly, they are the cytoskeletal structures. They form the cytoskeletal structures in the cytoplasm. That is, they act like a skeleton, or they provide the mechanical support to the cell and provide rigidity and a particular shape to the cell. Stiffness of the cell is maintained with the help of these cytoskeletal structures, that is the cisternate tubules and the vesicles. The second is they provide intracellular transport system. Intracellular transport system. They act like intracellular transport system. So within the cells, something is to be transported from one place to the other place. So these skeletal structures, they help in the transportation of the materials within the cell, not outside the cell, it is within the cell. Or if something is to be transported from the nuclear membrane towards the plasma membrane, or from plasma membrane towards the nuclear membrane. So this transport system, that is the endoplasmic reticulum, will act like the transporting system, which will take the materials move the materials from one region to another region within the cell. It is within the cell. Then these are involved in the formation of lysosomes and also Golgi complex. Golgi complex. They are also involved in the formation of cell plate formation during cell division, plant cell division. Then the RER is involved in protein synthesis, Then the SER is involved in lipid synthesis and metabolism. So they also provide various enzymes. They provide enzymes for synthesis of biomolecules. Various other biomolecules. Then they also store materials what they synthesize. Store materials that are synthesized by themselves. So these are some of the functions of the endoplasmic reticulum. Cytoskeletal structures I have explained, intracellular transport system I have explained. The lysosomes and the Golgi complex are formed out of these skeletal structures, that is the endoplasmic reticulum. They help in the formation of lysosomes and the Golgi complex. So during cell division, plant cell division, there are two steps, that is the karyokinesis and the cytokinesis. So karyokinesis is followed by the cytokinesis, and during the process of cytokinesis there is division of the cytoplasm, which takes place by the formation of a plate divides the cytoplasm into two pieces, so that one single cell will divide into two. So that process is brought about by this endoplasmic reticulum. The cell plate formation is brought about by the endoplasmic reticulum. Now the RER, that is the rough endoplasmic reticulum, since it is studded with ribosomes, They bring about protein synthesis. You might be knowing ribosomes are the site of protein synthesis. Henceforth, the rough endoplasmic reticulum is involved in protein synthesis. SER, smooth endoplasmic reticulum, they are involved in the synthesis of lipids and its metabolism. So, these endoplasmic reticulum, they also have various enzymes which are provided for the synthesis of various other biomolecules, maybe other amino acids, proteins, enzymes, proenzymes, vitamins, steroids, etc. So, they also help in storing all the materials that are synthesized by these endoplasmic reticulum are also stored by them only, and henceforth these are all the functions of endoplasmic reticulum. Now coming to the differences between the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Differences. So, SER and the RER. Ribosomes absent. Ribosomes absent. Here the ribosomes are present. Here the ribosomes are present. So, mostly they are in the form of vesicles and tubules. They are in the form of tubules and vesicles. Most of it is in the form of cisternae and a few tubules and vesicles. Few tubules and vesicles. So, SER is attached with the plasma lemma. They are attached with the plasma lemma, and almost these RER are embedded in the cytoplasm and they are connected to the nuclear membrane. They are connected to the nuclear membrane. So, SER is involved in the synthesis of glycogen, lipid, steroids, proteins, proenzymes, enzymes, etc. Vitamins also we can add, and here RER is involved in elaboration of the proteins and enzymes. Elaboration of the proteins and enzymes is brought about by the rough endoplasmic reticulum. So, with the help of diagram we have understood the endoplasmic reticulum and its components. They are there, cisternae, tubules and the vesicles. Most of the smooth endoplasmic reticulum, they are either in the form of tubules or in the form of vesicles, whereas RER, most of it is in the form of cisternae. So, you might have observed by writing the diagram of the cell, we had written endoplasmic reticulum which was attached to which was near the plasma membrane, and another group where it was attached to the nuclear membrane. So, SER is associated with the plasma lemma. They are very near to the plasma membrane and they are connected to the plasma membrane, whereas the RER, they are connected with the nuclear membrane. So, there might be some intracellular transport between the two which is carried out by these endoplasmic reticulum. So, that is about the endoplasmic reticulum. Next is Golgi complex. Golgi complex, or it is also called as Golgi Apparatus, or it is also called as Golgi body, or it is also called as dictyosomes. They are also referred as dictyosomes. So coming to the diagram, we'll write the diagram first and then we'll go to the explanation. So this would be called as the Golgi Apparatus. You can call this as the Golgi Apparatus. So they are also ultra microscopic. They are also ultra microscopic, and they are double membranous, interconnected. They are interconnected. Cisternae, vesicles, vacuoles, and tubules, which are embedded in the cytoplasm. The Golgi apparatus was discovered by a scientist called as Camillo Golgi. So these cisternae are almost disc-like. They are almost disc-like sacs. They are almost disc-like sacs, and so disc is like this. So disc-like sacs, they have a flattened surface like this. So they are disc-like and sac-like in its appearance, and they are placed one above the other to form a stack or a pile, and each pile or a stack consists of 8 to 10 cisternae. 8 to 10 cisternae or the sac-like structures are present in that stack. And the tubules, once again they are elongated tubules. They are elongated branched structures. Elongated branched structures. And the vesicles, they are spherical. And the vacuoles are also almost spherical structures where they contain in them granules and amorphous. So granules are something like the sugar-like granules are present, and something like amorphous, that is the talcum powder like things are present in the vacuoles. So these are the components which make the Golgi apparatus. Hence it is called as, because there are so many components, it is called as Golgi complex or the Golgi apparatus. It is called as Golgi apparatus. Now this cisternae, which are disc-like, have got two face like this. It has got two face. One is the convex phase and this is the concave phase. This convex phase is called as the cis phase, or it is also called as forming phase. It is also called as forming phase. And this phase, it is called as the trans phase. The concave phase is called as the trans phase, or it is also called as maturing phase. It is also called as the maturing phase. So mostly these dictyosomes or the Golgi complex, they are involved in secretion and packaging. They are mostly involved in secretion and packaging. They are secretory in nature and they are also helping in packaging. So what happens is, when some materials are to be packed up, so they fuse with the vesicles of the endoplasmic reticulum and they come and fuse with the cis phase, and from there they move to the maturing phase, and finally they are released into the cytoplasm. So any materials are to be packed. First, the endoplasmic reticulum, the materials are packed in the vesicles of the endoplasmic reticulum and they fuse with the cis phase of the endoplasmic Golgi complex and they are released from the maturing phase. So mostly these endoplasmic reticulum are concerned with secretion and packaging materials. So even the lysosomes, they are packed up with the help of these Golgi complex. So they are formed from the endoplasmic reticulum. Therefore, Golgi complex is formed from the endoplasmic reticulum, you can say. So this is the diagram showing the cisternae-like structures, the sac-like structures which are in the form of disc, which are placed one above the other, and you call them as the stacks. And these are the tubules, these are the vesicles, and these are the vacuoles. So all these together you call it as the Golgi complex or the Golgi apparatus. Now coming to the functions of this Golgi complex. So we have already told they are almost secretory and they act as packaging materials. They help in packaging and secretion. So they are involved in secretory activity. They are involved in secretory activity when during the synthesis of proteins, proenzymes, enzymes, proenzymes, lipids, etc. So mostly they are involved in the secretory activity during the synthesis of the various biomolecules, you can tell. The second is, it acts as a packing material. It acts like a packing material, which I have already explained. So it acts as a packaging material. Third is, it forms the sperm head. It forms the sperm head, which is called as the acrosome. So the acrosome is nothing but the head of the sperm. Sperm, they are the male gametes, and when you write the diagram of a sperm, it consists of a head-like part, the body, and a tail-like region. This is the middle piece, this is the head, and this is the tail. And in the head portion, there is a cap-like structure, which is the acrosome, which is formed by the Golgi complex. Next is, they are also involved in the synthesis of, they also help in the formation of lysosomes. So they also help in the formation of cell plate during cell division, during plant cell division, and these dictyosomes, they help in the formation of the contractile vacuoles. In some organisms like the freshwater forms like the amoeba. So these are some of the functions of the Golgi complex. They are involved in the secretory activity. They are involved in the secretion during the synthesis of biomolecules, and they act as the packaging material. They help in the formation of the acrosome or the sperm head. They also help in the synthesis of the formation of lysosomes and the formation of cell plate during plant cell division. They are also involved in the formation of the contractile vacuoles. So these are some of the functions of the Golgi complex or the dictyosomes. Now going to the next is lysosomes. So lysosomes were discovered by Christian D. Dewey. Lysosomes discovered by Christian D. Dewey, and these are almost spherical structures, almost membranous spherical structures. They are membranous spherical structures, almost spherical structures which contain lytic enzymes in them. This is more important. They contain lytic enzymes in them. So hydrolytic enzymes we call them as. So the lytic enzymes like the lipases, proteases, carbohydrases are nothing but the lytic enzymes. So the lytic enzymes, the breakdown of the lipids is brought about by the lipases, the breakdown of the proteins is brought about by the proteases, the carbohydrates are broken down by the carbohydrases. So these are the lysosomes which contain the lytic enzymes. And these lysosomes, they are of functionally they are of four types. One is the primary lysosomes. So the second is the secondary lysosomes. So autophagic lysosomes, and finally the residual bodies. See, lysosomes, since they have these lytic enzymes in them, sometimes the cell itself is burst out or completely destroyed because of these lytic enzymes. They destroy the cell in which it lives, and hence they are called as the suicidal bags. They are called as the suicidal bags or suicidal packets of the cell. They are referred as the suicidal bags or the suicidal packets of the cell. So the primary lysosomes. So these are lysosomes which contain these lytic enzymes in them. They contain the lytic enzymes in them. The secondary lysosomes are the lysosomes which attach to the food vacuole, In most of the primitive organisms, the food vacuole performs the digestion part, and this secondary lysosomes are associated with the food vacuole, and the lytic enzymes which are present in the lysosome will bring about the digestion of the lysis or the digestion of the food in the food vacuole. So they are associated with the food vacuole. Autophagic autophagic lysosomes. So these are also lysosomes which are which remove the unwanted waste materials, and throw it out of the cell. So they collect all the waste materials and they destroy it with the help of the lytic enzymes. They collect all the waste materials and they destroy the waste materials with the help of the lysosomes. Residual bodies almost the same, but they will throw off all the waste out of the cell by exocytosis or ephagy. The unwanted substances or it will also destroy some of the foreign substances that enter into the cell, because these lysosomes have the lytic cells. So these are the different types of lysosomes: primary lysosomes, the secondary lysosomes, the autophagic lysosomes, and the residual lysosomes. Now coming to the functions of these lysosomes. They bring about phagocytosis. I'll tell you what it is. They bring about the phagocytosis. Second is, so they break the organs during metamorphosis. I'll explain what it is. It brings about it helps in the dissolving of egg membranes during fertilization, and also it brings about the involution of uterus and mammary glands in case of mammals. I'll explain what it is. First one is, there are certain cells called as phagocytes which are formed out of the WBC cells, which contain these lysosomes in them, and the major function of the phagocytes is to identify any foreign substance entering into the tissue or the cell and destroy it. And it is destroyed with the help of the lytic enzymes which are present in the lysosomes. So that is one of the very important function of the lysosomes. The second one is, they break the organs during metamorphosis. That is, we can see metamorphosis is a process in which the young one is converted into an adult by undergoing several larval stages. I'll take the example of frog. We might have seen the frog tadpole which is having a tail, but the adult frog is not having a tail. What happened to the tail? So that tail has been completely removed by this process, where the entire tail has been removed by these lytic enzymes which are present in the lysosomes, and therefore tail is totally absent in the adult frog, whereas the tadpole has a tail. And another function of the lysosomes is these lysosomes, they are present in the form of lytic enzymes. They are present in the head of the sperm, in the acrosome, and they are released during the process of fertilization. The sperm head releases these lytic enzymes, so they dissolve the egg membranes and try to fuse into the egg nuclei, therefore bringing up the fertilization process. So these lytic enzymes of the lysosomes, they help in dissolving the egg membranes by the sperm during the process of fertilization. And another is involution of the uterus and mammary glands. See, what usually happens in mammals is, during the phase of pregnancy, there is enlargement of both the uterus and the mammary glands. Soon after the birth of the baby and the end of the lactation period, the mammary glands have to come back to their original size, or the uterus have to come back to their original size, because after undergoing enlargement during pregnancy, after the childbirth, they have to retain back their size. So all the cells are removed by the process of by these lytic enzymes. All the extra cells are removed by the lytic enzymes of the lysosomes. So that is the meaning of this particular statement. So these are the functions of the lysosomes. So thereby we will end up this video. Vacuoles will take in the next video. Thank you.